Good morning, good afternoon, and good evening to everyone joining from around the world. My name is Sabrina Welsh, and I'm so pleased to welcome you to the presentation of the 2020 Michelson Prizes. Our program will feature leading voices in the field, including our moderator, Pulitzer Prize winner Laurie Garrett, who will introduce the winners and preside over the presentation of awards. After the award ceremony, I'll welcome Dr. Dan Baruch, who will present his insights into COVID-19 vaccine development in the Global COVID Lab meeting. But first, I'd like to say a few words about our donor. The Michelson Prizes are highly competitive scientific awards given annually to innovative young investigators working to advance human immunology, vaccine discovery, and immunotherapy research across major global diseases. The Michelson Prizes are the vision of Dr. Gary Michelson and his wife, Alia. The Michelsons recognize that there is an imbalance in funding for early career investigators that is hindering progress in several areas of research. They launched the Michelson Prizes with the goal to both spotlight and support promising researchers who are utilizing unconventional approaches to advance vaccine and immunotherapy research. The Michelson Medical Research Foundation is committed to continue to explore ways to correct systemic problems with research funding and support cross-disciplinary efforts that bridge silos in scientific research to yield powerful outcomes. They also hope to inspire others to support similar funding initiatives targeting young researchers. Now in their third year, the Michelson Prizes are typically awarded at the Symposium for the Future of Vaccine Development. Unfortunately, due to the COVID pandemic, a face-to-face event isn't possible. But the pandemic itself highlights the importance of the work that today's winners are doing to advance our understanding of human immunology, to develop new vaccines. This year, the Michelson Prizes received hundreds of applications from around the world, representing diverse fields such as molecular biology, protein engineering, computer science, the microbiome, and nanotechnology. Now we'll hear a few words from our donor, Gary Michelson of the Michelson Medical Research Foundation.
Welcome, and thank you for joining us. One cannot leap a chasm in several small steps, but for far too long, it has been difficult, if not impossible, to find funding for high-risk, high-return medical research. The kind that leaps. The same could be said for disruptive research, the kind that disproves the status quo and turns over the apple cart. By far, the largest funder of medical research in this country is the federal government, whose own data proves that the system is rigged, with not most of the money, but rather nearly all, going to the old guard, with nothing left for the young, brilliant scientists that history has shown to be the very best at leaping. In fact, several studies of the ages of those who have won Nobel Prizes in the sciences have shown that the peak age for the intersection of scientific genius and creativity is under 35 years of age. My purpose in launching the Michelson Young Researcher Prizes was to correct both of those problems and to alter the trajectories of these scientists' lives. Particularly in academic centers, there has been this notion that the scientists there have been called to a higher purpose, science for science's sake. While I do believe in the importance of basic research, I believe even more in science for mankind's sake. I believe that breakthrough science, the purpose of the search in research, requires four things: mastery of the science, creativity, a sense of what is possible, and the courage to see it through. Today, I am very pleased that we are recognizing two researchers who have these qualities: Danika Hill and Michael Birnbaum. I would like to congratulate them and to wish them swift passage on their journey to someplace remarkable. Thank you.
Good morning, everybody, or afternoon or evening or whatever time zone you're in. Welcome to the Human Vaccines Project's Michelson Awards ceremony. There's going to be more than awards in store. Those of you that are tuning in to learn about immunology and COVID, welcome aboard. We really couldn't ask for a more perfect moment to think about why we need to support young, innovative immunologists right now. As you look around the landscape of what the crisis that we're all facing around the world with COVID-19, SARS-CoV-2, you can see a massive amount of uncertainty and confusion about how the human immune system is responding to this virus and what elements of the immune system actually matter, even including getting to our speaker that will be following the ceremony, talking about specifically how to develop a vaccine. If I can have the first slide, please. So, as I was saying before, nothing illustrates the need to have better understanding and innovative thinking in immunology than the quagmire and mess we're in right now with COVID-19. A mess that, of course, is exacerbated by political leadership that chooses to describe how the human body interacts with a virus in political terms rather than in biological terms. Imagining that it will simply poof, go away someday, or it's just a common cold, or it's a little worse than the flu, blah, 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 blah. In each case, next, we're dealing with, next slide, we're dealing with a tough situation in which the priorities are being set according to economic needs, political needs, and wishful thinking that isn't necessarily validated by the science. Sweden, of course, famously opted for trying to create a sense of herd immunity without anybody really knowing what exactly is herd immunity in this context. And in a pandemic, have we ever actually tackled a pandemic waiting for the herd, meaning human beings, to garner some form of immunity? Do we even know what that immunity is? Is it neutralizing antibodies? What is the nature of this immunity? Well, one thing we know is it didn't work in Sweden. Asking people to sort of take their own precautions, imagine their own ways of muddling through, has resulted in Sweden having the worst death rate per capita in that part of Europe and rivaling in the top 5 in the world, and in particular losing an older generation. Next. And when we look at the UK, where again they tried briefly to flirt with the idea of awaiting some sort of mythological herd immunity arising, the problems have been enormous. And actually today, the UK stands out as having the gravest both financial crisis and COVID crisis simultaneously in their region of the world. Next. Herd immunity, I mean, what in the world is it? The concept came out of veterinary and livestock science, and the idea was you saw rinderpest go through one part of your cattle herd, and you hoped that you got immunity before your entire cattle herd died of rinderpest. And we really don't know what is the critical point of herd immunity for human beings against a coronavirus. We've never entertained such a notion before. The sort of general feeling among most people that are in that overlapping area between epidemiology and immunology is that it's somewhere in the neighborhood of 65 to 70%. But there are people out there saying it could be as low as 20% of the population is immune, all the way up to you need more than 80%. So the truth is we have no consensus among immunologists or epidemiologists about just what percentage of the human population in any given location or planet-wide needs to have neutralizing antibodies against this virus or T-cell response of some kind in order for the herd, if you will, to be protected. Next, please. And the herd immunity question has arisen many times when New York City went through, of course, famously, the worst local epidemic so far in the history of this outbreak. And even with several months of circulation of virus throughout New York City, a very high death toll, at peak, it was 22.7% of the population had demonstrated neutralizing antibodies. And according to the health department leadership, this definitely does not constitute herd immunity. Vast majority of the population remains vulnerable. Next, please. Similarly, in Spain, which had one of the worst outbreaks in all of Europe, after the entire epidemic has calmed down and most of Spain has been able to return to something quote unquote normal, we're still looking at less than 5% population immunity to the virus. Next, please. In Wuhan, which of course is where this all began and had an enormous epidemic in December, January, and February. They're looking at a very low level of lasting immunity in the Wuhan population against this virus in any effective way. Next, please. Now, there are also anecdotes out there, and it's very hard to know whether any of these are valid and what exactly is going on here, but there are many such anecdotes of individuals who recovered from COVID and then apparently were reinfected. If this is true, then immunity, at least for some subset of the population, is not of any significance in terms of protecting the individual from infection. We need a lot more work to understand if any of these are real, and if they are, why is this happening? Next, please. One thing is very clear: the smart elements of the financial community are in strong agreement. Just last week, I was in a meeting with former Treasury Secretary Lawrence Summers, who was very much saying the same thing as you see here. We cannot salvage the global economy, which is in the worst shape it's been since 1929 in the Great Depression. And we cannot salvage any cluster or location of that economy around the world until we deal with this epidemic and get it under significant control so that people can return to work, return to school, return to life of some sort. Perhaps not 2019 life, but something better than the 2020 life we've been living for the last 3 or 4 months. And that's going to require an effective vaccine. Next, please. But we have to keep in mind that that vaccine must be made with great care. If there are any safety considerations, any doubts about its efficacy, a huge percentage of populations in the United States, and this is true in many countries around the world, a huge percentage are already inclined to refuse vaccination with a COVID vaccine. We have an uphill battle. If indeed herd immunity requires something in the neighborhood of north of 65% of the population having antibodies that are effective against the virus, then we already are on a losing side of this battle even before we have a vaccine. Next slide, please. And of course, we're already getting a lot of joking about what exactly is immunology and how well has the field performed in the face of COVID. Ed Yong is a well-known science writer for The Atlantic, and in his semi-mocking, lengthy essay, he basically says immunologists are a hodgepodge of witch doctors and artists playing their violin of ideas with the interferon on one side, the CD8 on the other, and a whole pile of acronyms and not really knowing what they're doing. And I hope that everybody understands that the Human Vaccines Project is dedicated to getting us past this sort of hand-waving Rube Goldberg approach to understanding how the human body responds. Next, please. But there's no doubt that we are in a very baffling position right now with this virus. And depending on what subset of the field of immunology you're in, you may be all about CD8 cells, you may be all about cytokines, you may be all about IgG, neutralizing antibody response. The truth is that what is happening in response to a virus that the human body has never seen before, that's in a class of viruses that the human body has never successfully fought off, meaning common colds that you can be reinfected with annually, we are looking at a very complicated response that we don't completely understand. Next slide, please. We have a lot of indications from various places around the world that the antibody response, when it does arise, is of short, limited duration. There have been studies in several places around the world that have shown that the majority of respondents who do muster significant neutralizing antibody response lose most or all of that response somewhere between 3 to 6 months. And of course, there are very few places where people have been infected for 6 months yet. So we have a very tiny Chinese study pool, next slide please, to look at for longer responses. Nevertheless, so far, the data is pointing to a troubling scenario in which people may muster, both asymptomatic carriers and those with acute disease, may muster an initial powerful antibody response, but it does wane in everybody and does so fairly quickly. Next slide, please. Longitudinal studies, especially in London, looking out over time, next slide, please, are showing us a similar picture of waning immunity. And even in London, where, as I said, they tried to experiment with the herd immunity, loosey-goosey approach to epidemic control, so that quite a lot of people were exposed to virus. Next slide, please. The neutralizing antibody response, initially robust, fell off in some people 23-fold within just 3 months, and was only stable and lasting in 17% of the London study group. Next slide, please. The waning of response seems to be seen universally if what you're looking at is IgG, IgM, IgA, your classic immunoglobulin responses. Next slide, please. And in this most recent, very confusing analysis in the New England Journal of Medicine, just published last week, you can see that actually the story is it's all over the place. There doesn't seem to be an easy way to look at what was the nature of the exposure of the individual, the nature of the response, symptomatically or asymptomatically, the individual had, and then 3 months later, how does that correlate with whether or not they still have an active, robust antibody response? Next slide, please. We are seeing some exciting possibilities that, at least in some isolated individuals, having been exposed in 2003 to the SARS epidemic virus, may have some cross-protective effect against the new SARS virus. And there may be some memory in the B cell compartment that is significant. Exactly, I'm using the word may all over the place, and every single sentence is going to be caveated because that's where we are right now. Next slide, please. We are beginning to see more and more evidence of strong T cell responses. And in fact, you could argue that one of the things that's going on right now in the field is that the sort of B cell immunoglobulin biased immunology subset sees this whole pandemic one way, and the T cell biased cellular response subset is seeing it quite a different way. From the perspective of the T cell response, it may be there is some kind of lasting memory. Next slide, please. And therefore, there's a whole new surge of saying, let's not get depressed. Just because there's no duration, next slide, doesn't mean that there's no actual immune response. You may not see neutralizing antibodies, but there's plenty of other things going on. Next slide, please. So how long does that all last? Next slide. If you're looking at the entire response of the immune system, every single factor, not just IgG. And as I said, what's happening now is a real push to convince the sort of consensus view that we should shift from focusing on immunoglobulins, thinking more about T cell responses, and looking at overlapping evidence of robust T cell responses to seasonal colds that are coronaviruses, and that perhaps having an overlapping effect for confrontation with COVID. Next, please. Again, we're seeing spikes in antibody titers and spikes in somehow correlated T cell responses. It's really quite confusing. Next slide, please. And as I say, there's a lot of evidence that the T cell response may be far more lasting and stable. Of course, what this means with vaccine production, well, I'm going to leave that to Dan to tell you, but that's a very confusing picture. Next slide, please. So if we are looking at T cells that are protective, then the question becomes, are they protective but perhaps play a role in enhancement or in some deleterious effect of the overall net immune response to SARS-CoV-2? There's so much here in these questions that goes right to the core of why the Human Vaccines Project exists, because in each aspect of these questions are unanswered fundamental issues about the human immune system that we've never really resolved. Next, please. As I said, there's increasing claims and evidence. All of this, mind you, when I say increasing claims, I'm talking week to week, day to day. That's the pace at which the science is unfolding. But there is increasing claims that a history of exposure to common colds, maybe some kind of an original sin issue similar to what is often talked about with influenza, that there's some kind of programming that may make a difference in terms of what is the nature of the immune response to SARS-CoV-2. We all know people who claim, I never get a cold. I've never had a cold. In my whole life, I've never had a cold. We hate those people. But there they are, and they make those claims. And if true, maybe there is something in the way their immune response sees coronaviruses. Next slide, please. And as I said, we're getting more and more evidence of how exactly the exposure occurs and what's the difference between the nature of lasting coronavirus responses to other coronavirus. How does that overlap with SARS-CoV-2? Next. And there may be multiple epitopes that are involved, epitopes that are triggering your IgG responses and those that are triggering T-cell responses. There are those who are already claiming to know exactly what we should all be looking for, but I would argue this is changing day by day. Next slide, please. And just to say that there are aspects of the immune system that are not usually talked about or looked at in most infectious disease contexts that are just beginning to come forward as, next slide please, important in the context of SARS and CoV-2, and of recognizing which aspects of the so-called S or spike protein are critical for triggering the immune response, and are there things that aren't even on the spike, other aspects of the virus itself that are crucial to triggering some aspect of the immune response. Next slide, please. So today we're focusing on two individuals who've done wonderful, innovative work that are an example of the kinds of fresh thinking, new ideas, new views, and new technologies that need to come to bear at a frenetic pace right now if we are indeed going to figure out how to conquer this virus and understand what it's doing to the human immune system. And I just would make one final plug for the basics of the Human Vaccine Project to say that, you know, Wayne Koff, who will be joining us in a second, has said many times, we still don't really understand some of the most basic elements of how the human immune system works and how it works with age, the difference between the neonate's immune system and the elderly individual's immune system. And so hopefully through the Michelson Prizes and other issues that HVP is working on, we'll gain greater insights in a timely manner. And with that, I would like to, do I hand off to Wayne? There, I see you in the corner. Wayne Koff.
We're going to see the two videos of the winners first, and then we'll have Wayne do a quick introduction.
Okay, sorry.
Thank you.
Meet the winners.
Thanks, Laurie.
Vaccines have tremendous power to improve health throughout our lives, but there are a range of infections that we still don't have vaccines for. So there are millions of lives that could be potentially saved each year if we had more vaccines. I'm studying a type of immune cell called a T follicular helper cell, and the body needs to generate these cells in response to a vaccine in order to produce long-lived antibodies that can then go on and protect us from infection. I was drawn to the fields of immunology because I've always been fascinated with how the body works. And the more I learned, the more I realized how much we didn't understand, and the immune system is still a big mystery. There are so many different pieces of the puzzle that need to work together in the immune system to keep us healthy. My research is trying to find what are the key pieces of that puzzle that we need to target through a vaccine in order to generate the best immune response possible. It's emerging that a key piece of that puzzle are the T follicular helper cells and the critical role that these cells play in generating good vaccine responses. Just as with everybody's DNA code, each person's immune system is unique. And this creates a real challenge when we design vaccines because they need to work for the majority of the population. The Michelson Prize comes at a critical point in my career. It will enable me to pursue my own research directions and enable me to follow up on some exciting discoveries that I've made. With the prize, I'll be able to apply some cutting-edge techniques to study hundreds of thousands of cells in molecular detail. And it's my hope that with this project, we can better understand what to pinpoint when we design vaccines in order to generate these T follicular helper cells that are so essential for a good vaccine responses.
My laboratory studies immune recognition. So in a word, I'm interested in knowing what the immune system sees, how that affects health and disease, and then what we can do about it. So the Michelson Prize for me is going to fund a new project idea that we have that would give us a way to systematically chart what the T-cell component of one's immune system sees during HIV. This is something that the field has been able to do T-cell by T-cell and person by person over the last 20 years or so. But we think we have a way that reuses viruses themselves in order to be able to chart this on a much more systematic level in order to understand not what's happening cell by cell or person by person, but hopefully on an entire population level. This will help us understand what are the more promising vaccine approaches and also understand why there's such a range in outcome of patients when they're infected with HIV, from those who respond quite well to treatments or naturally have a degree of control of disease, through to those who are quite susceptible throughout their lives. The Michelson Prize is going to really help catalyze this research in order to make sure that we're able to try some of our best ideas, which sometimes are hard to tell apart from some of our wildest ideas, and be able to do it quickly. Quick is also very important. And so being given money at this still relatively early stage of my career in order to go ahead and try these things, not worry about buying reagents, asking for help, hiring people is all going to be very helpful over the next year or two.
Okay. Wayne, are you on?
Is it my turn, Sabrina?
Yes.
Well, I just want to echo a couple of the points that our friend Laurie made. At the bottom line here is that we don't really understand how the human immune system works. I spent a lifetime in HIV vaccine development, and over the years we learned more and more and more about HIV. At the molecular level, And we made a lot of candidate HIV vaccines, and we still don't have an HIV vaccine. And I think the reason for that is that we don't really understand how to get the broadly neutralizing antibodies we want, how to get the broad and reactive of the T cells that we want. And then on the other side of the coin, if we learn how to do that, really how to get a long-lived protective response. And so that was really the origin of the Human Vaccines Project, which has come along at a time which is the golden age of human immunology. We have the tools here now to look at single cells in a way that we haven't ever been able to do before. We have a range of the arrays of the systems biology assays, all of the omics assays that we never were able to do before. And I think the most important issue is we have the AI and we have the machine learning, and which really has the ability, the aggregation of the enormous amount of data that we're generating to really understand how the immune system is really working. The epidemic that we have now, or the pandemic that we have now, has really highlighted this. And we have individuals that are asymptomatic. We have those that have mild disease. We have those that have severe disease, and we have those that are dying. And it's really likely that the baseline is predicting outcome, and we don't understand the baseline. And so the origin of the Michelson Prizes is really to address a number of these issues. When I first met the Michelsons now, I guess it's 3.5 years ago, four years ago, that Gary said to me that I'd like to really make an impact. I'd like to make an impact across a range of diseases. And we talked about how best to do that. And his idea was to launch a prize, and not to make a better HIV vaccine or a TB vaccine, etc., but not to make incremental progress, but to identify individuals out there that are really thinking outside the box, and to incentivize them. And so we began the prize 3 years ago. We got about 100 applications at that time, and we all, had 3 outstanding of the applicants who were each awarded a Michelson Prize. A year ago, we had a couple hundred applications, and again across the board, outside of the box, trans disease, all focused on one or another aspect of human immunology. And we awarded another set of the Michelson Prizes. In this year, we had even more of the applications. And the 2 of the winners who you're going to hear from in a minute just had incredibly outstanding applications and focused on 2 of the principal issues that have really challenged immunology over the years. And if we could understand how the follicular T helper cells work, if we can understand how the body, how the T cells really see a virus, it's going to open up doors across infectious diseases, across a range of other diseases. If you think about it, all of the cancer vaccines have failed, and even with an ongoing revolution in the cancer immunotherapy space, only a subset of people have gained any benefit. So there's a lot we don't understand. And as Gary pointed out in the video at the beginning of the webinar, his goal here is to really identify the needle in the haystack, if you will, and transform the fields. And so it takes a village to do the Michelson Prizes. It takes a number of people who are looking at the applications, reading the applications, prioritizing the applications. It's an intensive eight-month array of time. And at the end of the day, we have been lucky enough to identify two excellent winners in this year. So on behalf of the Human Vaccines Project, I want to congratulate the two of you. And I guess now, Sabrina, we're going to hear from each of the winners. Is that correct?
Yes, I'd like each of the winners to do their presentations. And if we've got time, we're running a bit behind, but I'd like to invite the participants attending to send your questions to Laurie using the chat function. If you can direct them to Laurie, she'll ask our winners 1 or 2 questions after their talks. Thank you. So Danika, I'm going to move to your slides now.
Thanks, Sabrina. I'm extremely humbled to be one of the Michelson Prize award winners for this year, and it's a real honor to join you today to talk about what the Michelson Prize will support me to research. Next slide, please. So during this SARS-CoV-2 pandemic, we're really experiencing an unprecedented pace at vaccine development. And I, for one, am optimistic that the hard work of immunologists around the world will pay off and it will see an effective vaccine against COVID-19 disease in the near future. However, for a range of viruses, pathogens, and bacteria, we still don't have effective vaccine solutions, despite having researched these pathogens for many decades. And for these complex pathogens, the tried and tested approach to vaccinology has really failed. So there's a need to come up with new ways to target these pathogens. Pathogens can be complex for a range of reasons. This can include genetic and antigenic variation, for example, for flu, or they can have thousands of protein-coding genes and diverse life cycle stages, like for parasites like malaria. But solutions for these pathogens will require a really in-depth understanding of how to elicit long-term protective immunity in humans, and importantly, to be able to find the antigens that can produce this response. An effective vaccine needs to work across the population and produce this long-term protective immunity. But really at the moment, we lack strategies to be able to design our vaccines in order to generate this response. Next slide, please. The majority of vaccines work by eliciting an antibody response. But antibody responses can really vary in their specificity, their affinity, and their longevity. The desired outcome of a vaccine would be high affinity and long-lived antibody, which is produced from long-lived plasma cells and affinity matured memory B cells. These cells form within the germinal center, which is a structure that forms within our lymph nodes after vaccination or infection. And it's within these germinal centers that a type of CD4 positive T cell, the T follicular helper cell, or TFH, is essential for supporting the formation, the maintenance, and the quality of the germinal center. These cells really dictate which B cells leave that germinal center as these long-lived plasma cells and germinal center derived memory B cells. Evidence from animal models has clearly shown that targeting the germinal center and TFH can induce long-lived immune responses and very specific memory. But we really lack strategies to be able to specifically engage these TFH cells in humans in order to generate a vaccine that can produce that desired outcome. Next slide, please. So these germinal centers are happening within our lymph nodes. And so that creates a challenge to study these cells in the context of human immunology and vaccination. But luckily, there's a population of TFH-like cells that circulates in our blood transiently after infection. Previous work that I've been involved in, we were able to demonstrate that these circulating TFH, or CTFH, are phenotypically related to cells within the germinal centers of the lymph node, in that they share a shared transcriptional profile, but they're also related at the level of T-cell receptor clonal relationships. So because these TFH cells are essential for supporting long-lived immunity, if we could find the antigens that elicit these cells, then these would have great potential to be turned into vaccines. Next slide, please. T cells detect antigen via their T cell receptor, or TCR. With the recent advancements in sequencing technology, we're able to really understand the human TCR repertoire in unprecedented detail. But the immense complexity of the human TCR repertoire, combined with the variability in human leukocyte antigen molecules, really creates a barrier to translate that sequence information into new vaccines. And at the moment, we're still not really able to assign antigen specificity just having that TCR sequence. But what we do know is that receptors that target the same epitope often share conserved sequence features. So we can use these identical or related sequences in the TCR and exploit these to really narrow the search for vaccine antigens. So in some of my previous work on CTFH cells responding to influenza and malaria vaccine, we detected that these common sequences occur with a high frequency between individuals. And so it's my goal to use these public clonotypes that we can find between individuals as a tool for precision vaccinology. Next slide, please. So my hypothesis is that the identification of common TCRs in CTFH cells will uncover important vaccine candidates for long-lived humoral immunity. And the features that distinguish my approach from what's existing in the field for T-cell epitope discovery is that by narrowing in and focusing on these CTFH cells, we're able to focus on a T-cell response that's directly linked to long-lived humoral immunity. And furthermore, if we focus on responses that are common between individuals at the outset, then this has potential to increase the likelihood of finding an antigen that can elicit good responses across the population. Next slide, please. So I plan to develop a vaccine antigen discovery platform in which we can take whole parasites, a cell lysate, a pool of peptides or proteins, and use this to re-stimulate immune cells, and then isolate activated T cells on the basis of activation markers. I plan to use single cell sequencing technologies to study the transcriptome of single T cells, but also to detect their alpha and beta chain paired sequences. Within that data set, I'm going to focus on T follicular helper cells and look at the TCR repertoire to try and identify those common patterns in receptors that are present between individuals. I'm then going to harness these sequences to generate reporter cell lines, which then I can use GFP as a reporter gene to screen for antigen specificity. So this approach will have trans-disease applicability, not only to discover antigens for complex and emerging pathogens, but really the plethora of non-infectious but immune conditions such as allergy and cancer, for which the targets of humoral immunity are still unknown. Next slide, please. I'm going to apply this method to studying immune responses to Group A Streptococcus. Strep A is among the top 10 leading causes of death related to infection and has a wide spectrum of diseases, from mild pharyngitis to life-threatening toxic shock syndrome, but also post-infectious autoimmune sequelae such as rheumatic heart disease. So there's a real need for a strep A vaccine, but progress in this area has really been hampered by a poor understanding of what are the targets of immunity. And so to this end, recently in Melbourne at the Murdoch Children's Research Institute, a human infection model has been established for Group A strep, where healthy volunteers are administered Group A strep to the back of the throat, and then they're treated with antibiotics at the onset of pharyngitis. So I'm going to be using blood samples collected in this model, which really allows a unique opportunity to study cellular immune responses against this pathogen in unprecedented detail. Thank you. Next slide. So this will be really a proof of concept study that meaningful vaccine antigen discovery is possible through targeting these TFH cells. I'll be using cellular immunology techniques, single cell sequencing, computational modeling, high throughput screening, and using some unique sample sets. And I'll be able to draw on the multidisciplinary expertise of a range of collaborators, both within Melbourne, at the University of Auckland, and in the UK. And just finally, I'd like to acknowledge Dr. Michelle Linterman at the Barouch Institute, in whose lab I generated a lot of the discoveries around TFH cells that supported this proposal. And thank you.
Oops. Okay. So I think we've got time for about 1 or 2 questions. Laurie, if you wanted to do that now, if you can unmute, please.
Great talk, very exciting. And you stimulated Dr. Kumar, who wants to know, obviously, wants you to leap right away to COVID model and to know if it's possible to look for TCR specifically for modeling COVID vaccines.
Yeah, that's a really interesting question. And actually something that I'm definitely pivoting towards and looking to apply this method to look at those T cell receptor patterns in COVID-19 patients. The interesting thing is some data out of the University of Melbourne has shown that in patients convalescent from a SARS-CoV-2 infection, that they induce a circulating TFH response. So this virus does seem to induce a germinal center response, and that these cells are antigen specific. So yeah, I think there could be some really interesting analysis looking at, do different groups of people induce the same types of T cell receptor responses?
But of course, that depends on knowing what the antigen is that you want to be focused on. Do you feel confident we know that for COVID?
I don't know that maybe we know the full picture, but you know, the coronaviruses are quite a lot simpler than say other complex pathogens where you've got hundreds of thousands of targets to choose from. Really, it's a handful, and I think we've got some nice data coming out about, you know, spike protein and potentially nucleocapsid. So I think by having a pretty targeted approach, say picking, you know, 4 or 5, we could have a pretty comprehensive understanding pretty quickly.
Terrific. And I think now we're going to turn to Michael Birnbaum and your presentation.
Great. Thank you, Laurie, for the introduction, and Wayne for your comments. And Danika, it's great to hear about your work. And apologies in advance if the fire inspector comes to my house. Hopefully that does not coincide, but it might. So I'm happy to tell you a little bit about the project, which I'm very excited was funded by the Human Vaccines Project at the Michelson Foundation, which is repertoire scale determination of T cell recognition. So I'm going to tell you a little bit about sort of the scope of the problem we're trying to solve, and then the tools we're building in order to try to solve it. So next slide. So I've spent my career interested in these molecules that are on your slide here on a truly a molecular level. So T cells do all of their recognizing of pathogens or cancer cells or your own cells in the case of autoimmunity via the T cell receptor. So each T cell has its own receptor, which binds to a peptide MHC molecule. So the T cell receptor here is in pink and blue, and the MHC is in green. And the MHC's entire job is to present short peptide fragments to the immune system for inspection. The result of this is wide-ranging and very powerful in that this is how the T cell the T cell repertoire coordinates the rest of the immune system in order to do all the various jobs it needs to do to protect you from disease. And in the case of CD8 killer T cells, it's literally what produces a life or death decision for the cell. So all of this is done under very exacting conditions. You can imagine that if this is wrong, that it is bad for you and your health. But this is still done with incredibly low affinity interactions. So 1 to 100 micromolar, this is on the range of say 1,000 to 100,000 fold lower affinities than you would normally expect for an antibody. But despite that, it's still incredibly sensitive, that just a handful of these targets on one cell would be enough to sufficiently activate a T cell. So next slide.
Sorry about that.
There we go. Awesome. So not only that, but your immune system has a numbers problem that it has to deal with as well. So, and that problem is that you can actually get an incredible amount of diversity in your immune receptor repertoire. So theoretically 10 to the 15, which is a giant number. In me or you, there's probably more like 10 to the 8 unique T cell receptors, which is still a giant number. And they're there tasked to recognize the possible combinations of antigens that might be there, which is on the order of 1 trillion that you could theoretically see in your life, with no real preconceived knowledge of which ones are going to be important to keep you alive versus which ones are things that you would never encounter. But despite that, we've known for a long time that if you have an antigen, if you have something that you would want to produce an immune response to, with some rules and exceptions, you're able to. So you're able to produce an immune response pretty much to anything. So that means that your immune response has to be very sensitive, very specific, but also have a degree of cross-reactivity. So every T cell can see in theory, on the range of say 50,000 to 100,000 antigens. And I've spent my career trying to understand how the rules of these systems work together. And I've also spent my career trying to build better tools in order to examine how these might interact. Next slide. And so we're interested in looking at this in a lot of contexts, but one of the most interesting to me is HIV and the concept of HIV elite controllers. So as I'm sure that you know, the HIV is still an incredibly impactful and harmful pandemic to the world, with just huge amounts of morbidity and mortality, particularly throughout South Asia, South America, and Africa. But there are a small cohort of patients who are able to naturally control this infection fairly well. So this is separated into progressors, who are on the left side of the slide, where the virus will eventually deplete T cells and have titer go up and take over, versus controllers, who are able to control HIV very similarly to the way that they would control any other virus. And this has been a well-known phenomenon for many years, but figuring out exactly what the immune system's seeing that separates these two phenomenon is something that is still very much under active research. And next slide. And one of the big challenges here is that the just degree of antigen diversity that's there is huge. So here, these squiggles on the right give you an idea of the genetic diversity that is present in all sorts of different viruses. So the small pixelated blob would be the equivalent of a typical yearly flu genetic diversity, which is roughly equal to the genetic diversity that you'll find in an HIV patient singularly. So one HIV patient will contain the same amount of just variation in their virus as the entire world would in flu, let alone the entire world in HIV. So it's a giant problem, and we want to be able to understand what's seen by T cells, and also how does the immune system tolerate these different mutations? What correlates with good outcome, both in terms of what antigens you see and how you see them, how you're able to cope with various escape mutation strategies. Next slide. And so we're interested in this on many scales, and the field of immunology has really been driven by technologies and tools to answer questions like these. So one possibility is you can start with one antigen that you care about, and you're able to find T cells, which is a tool that we've had as a field for on the range of, say, 25 years now. Next slide. There's also been a whole range of approaches that have been able to, say, take a T cell or a small number of T cells and ask what antigens they might be able to see. So this would be taking something where you have a response to a tumor or an infection, and you try to figure out why that's there. Next slide. But what I'm interested in is trying to build this as a connectivity map, right? Like in the end, we don't want to know one side of this or the other. We want to know how all of this is working together. We want to say we have T cells on one side and a whole host of possible things that could be seen on the other side. And we want to be able to know how that actually fits together to understand your entire immune response to a particular pathogen or to a tumor or to an autoimmune disease. And ideally do it in a way where we could do it not just one person, but do it with a tool that could be spread to the community as well. Next slide. So this is our idea for how to do it. And it's actually to repurpose HIV itself as an antigen discovery platform. So HIV is a lentivirus, and lentiviruses have been used as biomolecular tools for 20 plus years now. And like any virus, the cell entry or tropism is controlled by particular interactions on the surface of the virus and the surface of the cell that it targets. So this is, for example, why HIV infects helper T cells and not killer T cells. And these viruses also will encode into the cellular genome whatever is in the virus itself. And we know how to build big libraries of these viruses. So our idea, next slide, is what if we could control the infection of a virus like HIV to only infect antigen-specific T cells as a tool? What we would be doing then is having these viruses only infect a cell that they were able to see, and then leave some information in that cell about what allowed this to happen in the first place. So in this way, you would be able to have real control and a permanent mark on T cells that you're studying to understand what they recognized, and you'd be able to do this at very large scale. Next slide. So in work leading up to this proposal, we've built the tool that's able to do this. So what I'm showing you here on the left side is a engineered virus that encodes a T cell to express a fluorescent protein GFP. And so when that virus is put with the T cell that it's specific for, you get efficient entry and the cells begin expressing the fluorescent marker, where you get very good specificity on the other side. So if you put the virus with a cell that doesn't have the right antigen specificity, then you get very little to no infectivity. So this is the tool that we're able to use it. We're able to do all sorts of interesting targeting and tagging and gene therapy approaches this way. But the reason we actually built this, next slide, is to do this, is to basically build a library of thousands to tens of thousands of these peptide MHC displaying viruses, which we would be able to then take as a single tube, mix with T cells from a patient, and then sort away the T cells that are able to have a virus interact with it, infect that cell, and then analyze to understand both sides of my connectivity map in a single experiment, to be able to say, okay, here are the T cells that were recognized because we were able to sort them for a fluorescent marker. Here's the immune receptor that did the recognizing, and here's the target that they were able to see. And do this not cell by cell, but do it repertoire by repertoire instead. Next slide. And so our goals with this that we're going to be doing during the course of the award is to continue to build and validate our tool to work on library scale. So we're confident that we're going to be able to get this into the range of thousands of specificities, and use this with our collaborators at the Reagan Institute here in Cambridge to
try to really, on primary patient samples, understand what some of these differences are between HIV controllers and progressors, which has been done before, but been done much more by looking at anecdotal examples rather than be able to look as comprehensively as we're hoping to here. We're also expanding this tool for other diseases to try to build a more comprehensive look. So for example, because I'm sure that it's a question on your minds, in order to comprehensively look at all of the possible SARS-CoV-2 antigens that would be occurring in you, that works out to roughly 3,000 or so specificities per person, because our HLAs are polymorphic, your 3,000 are different than my 3,000. But that's a tool that we'd be able to build on comprehensive scale as a consequence of the work that we're doing in the lab. We're also building this with the hope that we would be able to say, construct libraries that, say, Danika could use and send them along. So have things that we could freeze down and without really any additional needed specific expertise, have it used by the community. And also build the databases where we would hopefully be able to use this computationally as well. So that's a look into what we're doing. And if there's any time, I'm happy to answer a question or two.
Well, I'm going to take the moderator's prerogative and ask you a question of my own. I think it's very exciting. And I assume that you're looking at, once you have the fluorescein or rhodamine markers or what have you on your T cells, you're looking at live cells. So you're using a fluorescence activated cell sorter, you're looking at live cells, you're able to then do perturbation or experimentation with those cells. What are you hoping to figure out when you get to that level and you've got libraries of live cells that you can trigger responses to? What are you hoping to see?
Yeah, so we're hoping to see a few things. So one is just on that full level, so that the fact that the cells are live is going to let us sort them away to understand, which ones are actually antigen specific. But then there's a question of not only what do you see, but how are you programmed? And so by sorting away the live cells, and there's some caveats here because we're using these viruses and we need to figure out how big of a perturbation that itself is. But there's a possibility that through single cell sequencing, the field has advanced far enough that we're going to be able to get in one experiment, what is your T cell receptor? What do you see? And also, which of the various immune cell programs are you wired with, to try to build that information as well. Just so really at any step of the perturbation, we'd be able to see. So we still have, it's early. We have a lot of work to do to get there, but that would be like the big picture goal for us.
So exciting. I mean, that would just be, I don't know if everybody watching this understands, but that could be a total game changer in how we go about trying to understand basic questions like, what is most important in the immune system? The amount of antigen you're exposed to, in other words, the sort of dose of virus that gets into you at a moment, or your genetic programming in your HLA, et cetera, or something very specific about that initial, dance between the first people on the dance floor, the first cells on the dance floor, and the antigens presented to them. It's very, very exciting.
Oh, it's a lot of fun to work on. A little more complicated fun with the diseases involved, but hopefully we could do both.
Well, my personal congratulations to both of you. I think you're on the path to some really important work and very exciting. And thanks to the Michelson family for putting up the support for you. I want to hand it back now to Sabrina for the next stage of our gathering.
Thanks, Laurie, and thanks to our winners and to Wayne Koff, our CEO, and thanks also to Gary and Alia Michelson for your support of the Michelson Prizes. We are thrilled to see these winners progress. I'm going to go ahead and switch slides so we can go into the Global COVID Lab meeting with Dr. Dan Baruch. Okay. And Dr. Baruch, I'm just going to start our introduction. If you can turn your video on, please, we'll begin the lab meeting. Hi.
Sure. Good morning, everyone.
Good morning. So once again, hi, my name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for today's webinar. Thanks to everyone who joined the presentation of the Michelson Prizes earlier today. Through the Michelson Prizes, we are committed to supporting and cultivating the next generation of young investigators who are advancing human immunology, vaccine discovery, and immunotherapy development across major global diseases. We're really proud and excited to see the winners move ahead with their projects, and we thought it would be a great way to bookend the Michelson event by presenting it alongside our global COVID lab meeting, which provides researchers with a forum to share their findings and innovative ideas with scientists from around the world. Today's speaker is one of the leaders in the COVID-19 field, Dr. Dan Baruch, who recently published an important study in Nature that gives us new clues into how a potential vaccine might work for COVID-19. Dr. Baruch is the director of the Center for Virology and Vaccine Research at Beth Israel Deaconess Medical Center and professor of medicine at Harvard Medical School. In addition, he is a key part of the Bill and Melinda Gates Foundation Collaboration for AIDS Vaccine Discovery, the National Institutes of Health Martin Delaney HIV Cure Collaboratory, and the Reagan Institute of MGH, MIT, and Harvard. Dr. Baruch's laboratory focuses on studying the immunology and virology of HIV infection and developing novel vaccine and eradication strategies. He has also applied his vaccine expertise to other infectious diseases, such as Zika virus, tuberculosis, and now COVID-19. He has advanced novel adenovirus vector-based HIV vaccine candidates from concept and design to preclinical testing, phase 1 and 2A clinical trials, and a large phase 2B efficacy trial with the Mosaic Ad26 on vaccine in sub-Saharan Africa. He has also pioneered the use of broadly neutralizing antibodies for HIV cure strategies, and a series of phase 1 clinical trials are currently underway. Dr. Baruch also led the world's first demonstration of Zika vaccine protection in preclinical studies and has launched a series of phase 1 Zika vaccine clinical trials. Dr. Baruch is board certified in internal medicine and infectious diseases, and he is committed to mentoring students, clinical fellows, research fellows, and junior faculty, and to providing clinical care to patients with infectious diseases. Just a quick note before we begin. The information presented today includes some pre-published data that is currently under peer review. At the request of the author, we ask that you not reproduce or disseminate the data presented. This session will be recorded and available on our website and our social media channels for you to review. In today's lab meeting, Dr. Baruch is going to talk about COVID-19 vaccine development. During the presentation, please send your questions to us using the Q&A function in Zoom. I'll ask our speaker a broad selection of your questions after he's finished his presentation. We'll have about 20 minutes for discussion, and I hope you'll take the opportunity to ask your questions about the data presented today. It is now my pleasure to introduce Dr. Dan Baruch from the Beth Israel Deaconess Medical Center.
Well, thank you, Sabrina. I'll try to share my slide. You cannot start screen share while other participant is sharing. So I think you have to.
So I just stopped my screen share. Okay. You should be able to grab the screen now.
Share.
Great.
Okay. Can you see my slide and can you hear me okay?
Yes, looks and sounds great. Thank you.
Okay, great. Well, thank you, everyone. Thank you for inviting me to give this week's Global COVID Lab meeting. I love the concept, by the way, of a global lab meeting. And so in the spirit of a lab meeting, then I hope this is informal. Feel free to ask questions either during my talk or at the end. But I hope that this is more discussion than a lecture. I was asked to present for about 20-25 minutes, and so there should be plenty of time for discussion. But before I start, let me also congratulate this year's Michelson Prize winners, Dr. Hill and Dr. Birnbaum, and for their incredible work to date, and I would say even more importantly, their promise for the future. So thank you both for your amazing work. What I'd like to do today is to give an update of some of our thoughts regarding COVID-19 vaccine development. I'll divide my time with some more general comments about the vaccine field and then some more specific comments about some of our recent data. So I think the COVID-19 pandemic needs no introduction. The devastating and surprising and extreme spread of this virus across the world, throughout our country, has been truly astonishing. And I think we've all been, sorry, let me X out of this. I think we've all really been humbled in what this virus has done to our world, our country, and how it's affected each and every one of us so personally. So why do we need a COVID-19 vaccine? And I think clearly the main reason is that it may be required, and I would even say it likely will be required, at least as part of a strategy to end this pandemic. I said this first in a similar Boston-wide mass CPR meeting in February of this year, or maybe it was March of this year, and I said a vaccine might be required to end the pandemic, and that was considered quite astonishing and newsworthy for some local media outlets. And for me, it was quite obvious at that time, but I think the spread of the virus over the last 6 months and the lack of ability to control this virus through public health measures, I think really necessitates the development of a vaccine. We started our vaccine work on January 10th, like many other groups in the world, when the sequence of the virus was published by the Chinese researchers. And at that point in time, I remember on that particular day, there were 41 cases reported in Wuhan, China, and 1 person had died. And we considered 41 cases to be quite concerning and quite a large number. Now, with more than 20 million cases diagnosed, and probably a vast number of more cases that have not been diagnosed, then the 41 initial cases in January seems truly like a drop in the bucket. But nevertheless, I think that this pandemic will likely need a vaccine. In addition, a COVID-19 vaccine could be important to control future outbreaks if this virus ends up being cyclical in some way. And regardless of the clinical utility of a vaccine, then vaccine work is an important contribution to basic science and our basic understanding of immunology, immunopathogenesis, and immune correlates of protection to a new human pathogen. So what are the key considerations for a COVID-19 vaccine? I think starting in early January, we actually thought that the development of a COVID-19 vaccine would be technically feasible. And the data to date over the last 6 months or 7 months has been reassuring. Unlike HIV that we have spent many years studying, and many people on this call I think have spent many years studying as well, for COVID-19, like for Zika in 2016, it was very clear from even the first description of cases that most people who get infected recover, and they appear to recover completely, at least in most cases. And that suggests that the human immune system has the intrinsic capacity to eliminate this virus. And we would extend and we would assume that there is at least some degree of protection against re-exposure. And while that hasn't been proven in humans, one of our early studies from a couple months ago, we proved that that was the case in monkeys. And so the ability of the human immune system to eliminate and to control the virus in at least the majority of humans suggests at least a degree of natural immunity. And that's critical because for HIV, for example, there is no natural immunity. That's one of the reasons why the development of an HIV vaccine has proven so difficult. In addition, unlike HIV, for which there is vast sequence diversity, then for COVID-19, there is only limited strain diversity. There have been several mutations developed that have been characterized, one particular one that appears to have largely taken over the global population, but that's not a knockout for vaccines that were developed of the original sequence. So there is some sequence variation, and there will surely be more sequence mutations that arise. But given the extensive spread of the virus, there is still a relatively limited amount of strain diversity. And thirdly, we're quite certain that we know what the key antibody target is, which is the coronavirus spike protein. That's based on years of work of SARS and MERS and common cold coronaviruses, as well as quite extensive data now over this year for SARS-CoV-2. But there are, of course, technical concerns for a COVID-19 vaccine. For example, there's the possibility of future mutations. And if there's a mutation that results in escape from the neutralizing antibodies raised by the original sequence, then it is theoretically possible that a variant could emerge that would essentially escape from all vaccines currently under development simultaneously. There's also a theoretical concern of disease enhancement for any coronavirus, and that's really based on not data from SARS-CoV-2, but data from the original SARS, in which there was several animal studies that showed that particular vaccines that raised, in some cases, suboptimal immune responses led to a worsening of respiratory disease upon challenge. That was never seen in humans, and to the best of my knowledge has not been seen in animals or humans for SARS-CoV-2, but nevertheless is a topic that researchers need to be very attuned to. And thirdly, the question of durability of protection. There is data in humans that the durability of antibody responses to common cold coronaviruses, as well as to SARS-CoV-2, may be limited in nature. It may not give the lifelong immunity that we would like to see from some other viral pathogens. And so therefore, the durability of protection of a vaccine remains an open question. And to the best of my knowledge, there have not been durability studies with any of the vaccines that are under development in either animal models or human clinical trials yet, for the simple reason that durability studies take time. So there are currently over 100 vaccine candidates under development, according to the WHO, and depending on how you want to count it, maybe even over 200 vaccine candidates. They span many different platforms, including DNA vaccines, RNA vaccines, purified adjuvanted protein vaccines, with or without delivery platforms, recombinant viral vector-based vaccines, as well as inactivated viruses, as well as some other cell-based strategies. The first vaccine to reach clinical trials was the RNA vaccine produced by the biotech company called Moderna. And I think that has really shown a lot of exciting news for that vaccine technology. But the timeline to start clinical trials and even the timeline to show efficacy in a clinical trial, which has not been achieved yet, is actually not the most relevant timeline in my opinion. The most relevant timeline for a COVID-19 vaccine is a timeline to produce and deploy billions of vaccine doses. In other words, it's not the timeline to show efficacy or even approval of a vaccine that's relevant. It's the timeline needed for the full end-to-end process of delivery of a vaccine to vast numbers of the human population in the world. And that's the relevant timeline. So people always ask, when will there be a vaccine available? And of course, it's impossible to predict the future. But if all goes well with the phase 3 trials that are currently underway, and if everything goes perfectly from this point forward, it is possible that there could be a vaccine or perhaps several vaccines available for emergency use authorization as early as this winter. Probably not for general population use until 2021. But those timelines are the optimistic timelines, and those basically are assuming that everything goes absolutely perfectly from this point forward. And as we know in science and medicine, it is not always the case that everything goes perfectly. So the question that has been debated a lot in the scientific circles as well as in the public is how to accelerate vaccine development. Vaccine development for a new pathogen that has never been seen before in humans usually takes many years or even decades. And so the attempt to develop a COVID-19 vaccine, or rather I should say vaccines, plural, in just 1 year would be truly unprecedented and essentially much, much faster than ever before in history. So how can this be done? And basically, there's several strategies for how vaccine development can be accelerated. First, using vaccine platforms that have already been tested for other pathogens. And so there does not need to be extensive, or in some cases, any preclinical toxicology work, and early dose finding in humans can be very rapid or even not relevant at all. And probably most importantly for vaccine developers, Many of them are performing development activities in parallel rather than in series. So let me give you one example of that, many, in fact, I would say all of the lead vaccine developers are mass producing their vaccines right now, even before demonstration of clinical efficacy. I would say that was previously would have been unheard of in the pharmaceutical industry, to take the financial risk of mass production of millions or even billions of doses of something before you know at all whether it is safe or effective at all. Because clearly, if it fails for any one reason or another, then all those millions or billions of doses will literally just be thrown out, and there would be a huge financial loss. So essentially, there is an ability to go much, much faster than usual by a high level of tolerance of programmatic and financial risk. Now, clearly that's offset by massive US government support for the different vaccine companies, but nevertheless, it's a financial risk, I would say, of not just the companies, but of the taxpayers as a whole. But importantly, this can be accelerated through no compromise and no risk to patient safety. So there is a high tolerance for financial risk, but no tolerance for patient safety risk. In addition, for acceleration of vaccine development, it's very important to move vaccines forward that have the capacity for mass production and distribution, and not a niche vaccine that might be hugely effective, but might only be able to be produced at small quantities. Because the goal is not just to protect the individuals who get vaccinated, the goal is to have enough people vaccinated to achieve herd immunity to actually end the pandemic. And the power of vaccines is not just to help the individuals who receive them, but to help society as a whole. So our own work on vaccines have really centered on viral vector-based vaccines. And one of the viral vectors that we have developed and have invested in over the years is an attenuated common cold virus called adenovirus serotype 26. We originally cloned and vectorized Ad26 in a Journal of Virology paper in 2007. And then we collaborated with a small biotech company in Holland called Crucell in the first development of this vector platform for our HIV vaccine program. And so the HIV vaccine program clearly has catalyzed our ability to do this work for other pathogens. And there would simply not be the Ad26 J&J program if there wasn't our HIV vaccine program over the last 15 years. I think we can say that with a fact. So there's a very clear synergy between HIV vaccine research and COVID vaccine research, not just for our program, but actually for many of the programs. So we initially brought Ad26 forward in a first-in-human study for HIV around 2009, 2010. And since then, Crucell was purchased by a large pharmaceutical company, J&J, and they developed the Ad26 platform at industrial scale. And they've developed vaccines, not just moving the HIV vaccine program, but we've also developed a Zika vaccine in collaboration with J&J based on Ad26. And J&J's developed Ad26 vaccines for Ebola and RSV. And last month was actually a big step forward in that the first Ad26 vaccine has been licensed for clinical use in Europe by the EMEA, and that's the Ad26 Ebola virus vaccine. And that was based on large-scale clinical trials. And two other features of Ad26 that are useful for a COVID-19 vaccine is that there is already quite a bit of experience with Ad26 vectors in humans. It's been proven to be safe and immunogenic in over 80,000 people to date, and I think this number is actually 90,000 now. The majority for the Ebola program, in which the Ad26 Ebola program has been deployed in Central Africa as a public health tool to try to quell the Ebola outbreak. And so we know that it's immunogenic even in areas of the world that have the highest levels of Ad26 baseline immunity, whose titers are generally quite low and are non-suppressive. But also we know that Ad26 vectors is an industrial platform that has already proven mass production capacity and has already been manufactured for the Ebola program at millions of doses. It's never been made at billions of doses, but it's proven to be mass producible at the level of millions of doses and global distribution. So we think those are important downstream attributes. So The Ad26 program for this vaccine really started on January 10th when the virus sequence was released. And we started working on the sequence that day, like many other groups, doing sequence alignments with SARS and MERS. And by Monday, January 13th, we developed synthetic genes that we ordered to design vaccine constructs. By the end of January, we had an agreement to work with J&J on this vaccine. They were the natural industry partner, given our work with them for other vaccines, Ad26 based. By February, we had immunized mice as well as rhesus macaques. And also in February, we developed viral challenge stocks. So it was quite amazing in the context of the epidemic unfolding outside our door. We were basically setting up all of the different systems in terms of viral challenge stocks, immunologic and virologic assays, small and large animal models, as well as vaccine immunogens. So in March, we basically validated the animal models. Particularly small animal models such as ferrets and hamsters, as well as large animal models such as rhesus and cynomolgus macaques. And I'll touch briefly on the two papers that we published in May demonstrating proof of concept of natural immunity and vaccine immunity. And then about 2 weeks ago, we published the immunogenicity and protective efficacy of the leading Ad26-based vaccine in non-human primates. And also last month was the initiation of our phase 1/2 clinical trials. And if all goes well, then next month, about a month from now, we should be in a position to initiate a 30,000 person phase 3 efficacy trial in collaboration with J&J and the NIH-sponsored co-VPN network to test the efficacy of this vaccine. So if all goes well, then our hope is that this vaccine could be available for emergency use authorization in early 2021. In March of this year, then J&J made a commitment to produce and manufacture at least 1 billion doses of the vaccine for worldwide use, and also importantly to produce and distribute this vaccine on a nonprofit basis. So clearly there's many other vaccines in development, and just for an overview of the field, the first vaccines that started phase 3 efficacy trials in the US were the Moderna RNA vaccine and the Pfizer RNA vaccine that started about 2 weeks ago. The expectation is the AstraZeneca Oxford ChAdOx1 vaccine will start in an efficacy trial in the US in the next week or two, and it's already in efficacy trials in the UK and in Brazil. And the hope, if the phase 1/2 studies are successful and rapidly generate the data, then the Ad26 vaccine should start a phase 3 efficacy trial in next month, and not soon thereafter will be the Novavax protein and probably also a Sanofi protein vaccine. There are also the trials of the inactivated virus vaccines produced by the Chinese groups as well, which is not shown on this slide. So 2 critical questions for COVID-19 vaccine development that we thought were important to answer as quickly as possible is there evidence of natural protective immunity? In other words, will individuals who recover from COVID-19 be protected against re-exposure? A critical question that's the fundamental concept for vaccines. And secondly, more germane to vaccines, is there evidence of vaccine-induced immunity? And if so, what are the immune correlates of protection? These are clearly questions that need to be answered in humans and require large-scale clinical trials to get these answers. We decided to address these in the rhesus macaque model to generate early animal model proof of concept data. And so these 2 papers were published in May of this year, at the end of May, and I think established the proof of concept for both of these questions. Just to show you one slide from the model development and re-challenge paper, here we infected rhesus macaques with SARS-CoV-2 by the intranasal and intratracheal routes, and we looked at virus in either lung or nasal swabs. Here is virus in bronchoalveolar lavage in lung. After the initial challenge, 9 out of 9 animals get infected, peak viral loads of about 10 to the 6, 10 to the 7 copies per mil of BAL fluid on day 2. And then they generally resolve their infection in about 2 weeks. we showed that these animals also developed humoral and cellular immune responses. And then if we re-challenge them with a second dose of virus, then they're protected. A couple animals have a very low blip of virus. In general, we think this is probably non-replicating virus that we sorted out using the subgenomic RNA assay, but the protection was astonishingly good. So at least in the short term, and we're not saying anything about durability here, but at least in the short term, there does appear to be very substantial natural protective immunity, at least in this animal model. We also performed an early DNA vaccine study. Now, these DNA vaccines are not the vaccines that we or others are taking into the clinic. These were purely research tools to get an early look on the concept of vaccine protective immunity and immune correlates of protection. So this was purely a basic research study, not linked to any clinical product, but rather a way of learning quickly and early about vaccine protection. And so we looked at a series of different spike protein variants, and there were differences among them. The one called S is the full-length spike, which did pretty much the best, although some others were quite similar. And we showed that 2 injections of this DNA vaccine did a pretty good job of reducing viral loads in the lung as well as in the nasal swabs. So this was evidence of vaccine protection both in the lower airways as well as in the upper airways. And the reduction of virus was 3 to 3.5 logs, with about half the animals showing no virus following challenge. So this was early evidence of vaccine immunity in macaques. In this study, we actually had a large number of animals, but we also had some DNA vaccines that worked, moderately well, not as well as the Ad26 vaccines, which I'll talk about in a minute. But some of them worked moderately well, and others actually worked very poorly, some of them in which the antigen was much less immunogenic. So the variability in the outcome was actually very good for an initial look at immune correlates. And we looked at many different immune correlate parameters, but the neutralizing antibody response induced by the vaccine was the strongest correlate of protection, shown here. So basically, the higher the neutralizing antibody response induced by the vaccine, the lower the viral loads in the lung. So this gave some early indications that neutralizing antibody titers might be a good biomarker for protection. And then 2 weeks ago, we published the first experiment that we did with Ad26-based vaccines. So I'll spend just a few minutes on data from this paper. We produced Ad26-based vectors in collaboration with our colleagues at J&J, expressing 7 different spike protein variations. We looked at 2 different promoters, a TPA promoter as well as the wild-type promoter. We looked at full-length spike, as well as a spike that's a deletion of the cytoplasmic tail called DCT, as well as a soluble ectodomain with a fibrotin tag shown as the open square. And then we looked at native sequences as well as sequences that have been modified according to the mutations that Barney Graham and Jason McClellan and others have described over the years for MERS, SARS, as well as SARS-CoV-2 that include a mutation of the furin cleavage site shown by the X between S1 and S2, and 2 proline mutations in S2, which are the 2 vertical arrows. And I'll just say up front that the vaccine that did the best is the one on the bottom, which is called, in this nomenclature, S.pp, which is the full-length spike with the native promoter and the diproline stabilizing mutations, as well as the furin cleavage site. So the study that we did involved 52 rhesus macaques. They were 10 to 12 years old, so they were on the older side, mixed gender. We decided to do a single shot vaccination because advectors are one of the few vaccine modalities in which there is substantial immunogenicity after 1 shot. Now, I'll say up front that the responses also are very much boostable. We've seen that with our HIV as well as our Zika program in humans. So we would expect a second shot to increase neutralizing antibody titers by a log to a log and a half. But the ad platform is immunogenic enough to give substantial immune responses after a single shot. So we decided to look at the immunogenicity and the protective efficacy of a single shot vaccine. That doesn't necessarily exclude that we might want to use a 2-shot vaccine in the clinic. So at week 0, we performed a single immunization with 10 to the 11th viral particles of Ad26 vectors expressing one of the 7 different spike protein variants that I described in the previous slide. These were 32 animals, so we had 4 to 6 animals per group that got each of the vaccines, and we had a large group of 20 sham controls. At week 6, we then performed a challenge with 10 to the 5 TCID50 of SARS-CoV-2 by the combination of the intranasal and intratracheal routes. So here are binding antibody responses after a single shot of the Ad26 vectors. At week 0, the baseline, there is some level of background. We think that might be cross-reactivity to some native primate coronaviruses or just an assay background. But clearly there was immunogenicity in all the animals at week 2 and week 4. The responses are higher at week 4 than week 2. And the highest responses were really in the 2 groups that had the full-length spike and the stabilized mutations with either the TPA promoter or the wild-type promoter. Here we look at neutralizing antibody responses by our own in-house luciferase-based pseudovirus neutralization assay. And again, there was no background neutralizing titers at week 0, and there were neutralizing titers in most of the animals at week 2 and in all of the vaccinated animals at week 4. And here, there was a clear winner of the S.pp vaccine that had neutralizing antibody titers after a single shot in the high 100 range. In our assay, this was about 4-fold higher, or 4 or 5-fold higher than the neutralizing antibody titers in a cohort of convalescent monkeys that were protected against rechallenge. Actually, the first paper that I showed you, that those were those monkeys, as well as a cohort of 29 patients with moderate to severe disease from the Brigham and Women's Hospital, a convalescent human cohort. In addition to traditional binding and neutralizing antibodies, we worked with Galit Alter and her team to look at different aspects of antibody profiling and function. And here we look at the different vaccines on a series of different both spike-specific and RBD-specific responses, various isotypes from IgG1 all the way up to IgG3 and IgA and IgM and IgA, as well as a FC receptor binding characteristics, all in red, as well as different functions in blue, such as antibody-dependent neutrophil phagocytosis, antibody-dependent cellular phagocytosis, antibody-dependent complement deposition, and antibody-dependent NK cell activation in blue. And you can see in these radar plots, the S.pp vaccine gave the broadest responses, both the binding responses and the functional responses compared with the other vaccines. The second place vaccine was the TPA SPP vaccine that also showed quite a large profile of responses. And then this portfolio of responses can be visualized in a principal component analysis shown on the right, really just to look for clustering. And most of these actually cluster with each other, with the full-length spike S as well as the SPP vaccines sort of on the extreme ends of this. We also looked at cellular immune responses, and here we show that essentially all of the SPP vaccinated animals generated interferon gamma responses of a moderate magnitude, and essentially no IL-4 responses, as you would expect for a viral vector-based vaccine. So as we would have expected, the viral vector-based vaccine gives essentially a pure Th1 response with essentially no evidence of Th2 responses. We then challenged these animals by the intratracheal and intranasal routes. And we measured, for protection, we measured viral loads in the lung in bronchoalveolar lavage taken at multiple time points following challenge, as well as in the nose in nasal swabs. So here we show subgenomic RNA copies per ml in bronchoalveolar lavage in the sham controls, in a large group of 20 controls to get a statistically robust data set, 100% of animals, so 20 out of 20 animals were infected and had median subgenomic RNA viral loads of about 4.5 logs up to about 6.5 logs. And in general, these resolve by between day 7 and 14. In the group that received the optimal vaccine, the S.pp vaccine, none of these animals had any virus in the lungs at any time point, suggestive of complete protection in the lungs. In the other 6 vaccine groups that had 4 to 6 animals, then there was also substantial protection in all the vaccine groups. In most of the vaccine groups, there were 0, 1, or 2 breakthroughs, but these breakthroughs generally were at lower magnitude and shorter duration than seen in the controls. So there was substantial protection with all of the vaccines, but the best protection in the SPP vaccine that had the highest neutralizing antibody responses. Viral loads in nasal swabs are shown here. These are subgenomic RNA copies per swab in the sham controls on the left, and in the SPP vaccinated animals on the right. Historically, for essentially all vaccine studies that have been published, protection in the upper airways has been a higher bar and less successful than protection in the lower airways. Here we see virus in nasal swabs at moderate to high levels, a median of 5.5 logs in 100% of the animals, so 20 out of 20 animals had virus in nasal swabs. In the SPP vaccinated animals, 5 out of 6 animals showed no virus in the upper airways, and 1 out of 6 animals had a low amount of virus seen in 1 animal. So also very good protection in the upper airways. We also saw a variable level of protection in the other 6 vaccines, in which there were 1 to 3 breakthroughs in each case. And these breakthroughs generally were lower and tighter, although as you can see, the protection in nasal swabs is not as good as protection in the BAL. So here's a summary of protective efficacy of these 7 different vaccines compared to the placebo group, the sham vaccinated group. And you can see that all of these vaccines show substantial protection in the lungs, and most of these vaccines show protection in nasal swabs. But the best protection was seen in the group on the far right, the S.pp vaccine, which was complete in the lower airways and was near complete in the upper airways. We also, because of the size of the study and also the variation in the outcome, so for correlates analysis, you need a large enough sample set and you need a variation in outcome, because if you only test one vaccine that works perfectly, then actually there's nothing to correlate and there's no insights that can be gleaned in terms of correlates of protection. Here, we did have a situation where some of the vaccines worked very, very well, and some of the vaccines actually didn't work very well. And so if we pooled everything together and we looked at all the animals all at once, then we actually had a robust data set that supported a correlates analysis. And here we show that the log neutralizing antibody titer on the x-axis inversely correlates with peak log subgenomic RNA copies per ml in BAL. So essentially, the neutralizing antibody titer correlates with protection with a very good p-value and sort of a moderate r-value. We also looked at many other correlates, and because many of the antibody parameters correlated with each other, then of course there were many different correlates in this study. Here we also show that the neutralizing antibody titer raised by the vaccine also correlates with protection in nasal swabs. So also in collaboration with Galit and colleagues, we also looked more broadly at the correlates of protection in both BAL and nasal swabs. And many of these antibody parameters correlated because many of them were correlated with each other. But if we look at correlates in a different way, and we look at what antibody parameters can separate the animals that were completely versus incompletely protected, there were about half the animals in this larger study showed complete protection, and the other half of the animals showed partial protection. And the parameter that best discriminated the group that had complete versus partial protection was the neutralizing antibody titer shown on the left graph on the bottom. But also other parameters also discriminated between these two groups. The second best correlate was the antibody-dependent NK cell activation, shown in the middle, and the third was antibody-dependent cellular phagocytosis. So we think this is a situation where the neutralizing function is likely the principal correlate of protection, but that other antibody functions might also contribute, probably to a lesser extent. We also looked at post-challenge immune responses to get a sense as to the degree of protection. And we saw something very interesting. In the sham controls shown on the left, they were negative for neutralizing titers at baseline prior to challenge. And by day 14 following challenge, they all had developed neutralizing titers of around 100, which is what we saw in our previous study. In the SPP vaccinated animals, these titers were higher than the convalescent animals to start with, but then there was actually no expansion of these titers post-challenge. So it really was like these animals' immune systems actually didn't even see the virus. And so this is actually, I think, probably the strongest evidence for complete protection in these animals. I'm specifically not using the word sterilizing immunity because I'm actually not sure what that word means. But the protection was essentially complete by any virologic or immunologic measure that we can look at in the lungs. That was not the case for the other vaccine groups. Here, most of the other vaccine groups actually did show an anamnestic response between pre-challenge and 2 weeks post-challenge, shown here. A couple animals in a couple of the groups, a couple animals in the TPA-SPP group and the SDTMPP group actually did not show expansion of antibody responses. But all of the other animals did show anamnestic responses, which is what exactly you would imagine if there was partial protection and essentially incomplete, if there was partial protection and then virus was rapidly controlled through immunologic anamnestic responses. We also looked at T-cell responses in these animals, and here we show T-cell responses 2 weeks following challenge in the sham control group on the left and in the SPP vaccinated group on the right. CD8 responses on the top and CD4 responses on the bottom. You can see that the sham controls developed robust T-cell responses, CD8 and CD4, against multiple different COVID proteins. The spike, S1, S2, nucleocapsid, and then non-structural protein 6, 7, and 8, which is the pools we looked at, which is what you would expect from a viral infection to generate broad T-cell responses against multiple antigens. In contrast, in the SPP vaccinated animals, there were low levels of responses to the vaccine antigen spike, but there was no evidence of broadening of T-cell responses to non-spike antigens. In other words, there was no development of T-cell responses to antigens that were not in the vaccine, adding to the evidence of essentially a very good level of protection. So we would conclude that there was a very low level there was at most an exceedingly low level of virus replication, if any at all, in these animals. So briefly to conclude, the Ad26 SPP vaccination resulted in complete protection in the lung in 6 out of 6 animals and near complete protection in the upper respiratory tract in 5 out of 6 animals. The lack of anamnestic responses suggests minimal to no virus replication in these animals post-challenge. We believe that a single-shot vaccine for COVID-19 would have major advantages over a 2-shot vaccine for mass immunization and pandemic control. However, a second shot of homologous Ad26 vectors in humans, we would expect to increase neutralizing antibody titers by more than 10-fold, and we saw that for our HIV program as well as our Zika program. So we actually think there is merit in testing a 2-shot vaccine as well for potency. So the current clinical trials actually are testing both a single-shot vaccine regimen and a 2-shot vaccine regimen. We saw neutralizing antibody titers as the strongest correlate of protective efficacy, although many other antibody parameters also correlated. And this was consistent with our prior DNA vaccine study. And so if you take this Ad26 study and our DNA study and you put them together, that's a total of 87 monkeys. And so we're actually starting to talk about numbers that are not so small anymore. And then the development plans for the Ad26 SPP vaccine, which has now become known as Ad26-CoV2S, is a phase 1/2 clinical trial that's ongoing now. And if the week 4 immunogenicity and safety data looks promising, then this will trigger the initiation of a large phase 3 clinical efficacy study in about a month's time with J&J and the co-VPN and, of course, many other partners. So I'll just end with a slide of a couple broad picture perspectives to try to tie things together. Vaccine development for COVID-19 is proceeding faster than for any other pathogen in history. Multiple vaccines, in our opinion at least, should be pursued in parallel, since it is not yet clear which will be the most safe, protective, developable, and deployable vaccine, as well as durable vaccine. We believe that preclinical challenge models can assist in the clinical development of these vaccines by providing useful insights into vaccine potency, efficacy, and immune correlates. And finally, it's important to realize that there are 7 billion people in this world, and no single vaccine manufacturer or company or supplier could possibly satisfy this global demand. So therefore, we actually need multiple vaccines to be successful. So this is not a race of one vaccine developer against another vaccine developer. This is a collaborative global effort to develop multiple vaccines that are going to be needed for the world to end this pandemic. I'll stop there. Thank a large number of people who are involved in the AD26 study at Beth Israel Deaconess Medical Center, at J&J, at BioQual, University of North Carolina, Children's Hospital, the Reagan Institute, and MIT. And this is a picture of our lab last fall and happier times when we were able to get together like this. And we hope that with a vaccine, we might be able to do this again soon. I'll stop there and take any questions.
Dr. Baruch, thank you so much. That was really exciting data. We've received a ton of questions, so I'm going to get right into it. Let's start on the development side and the administration of the vaccines. So upper airway infection seems pretty key here, which implies the best administration route for vaccine might be a nasal mist. Would this be the intent of J&J? Are they going for a nasal vaccination administration for this candidate?
So the current clinical trials are an intramuscular vaccine. We are considering doing some preclinical testing with nasal administration. So far, we have not had a lot of success delivering a replication incompetent vector on mucosal surfaces. We tested that in our HIV program for AD26, and actually the best mucosal immune responses, we got stronger mucosal immune responses with an intramuscularly administered vaccine compared with a mucosally administered vaccine. But admittedly, we were looking at mucosal responses in the gut. And so we haven't actually done that experiment for the nasal mucosa. And that's an experiment that clearly needs to be done.
Yeah, sure. And in terms of determining the duration of immunity, it seems like there's a rush to get these vaccines to phase 3 and to get them into the population. These phase 3 trials may be cut short. What is the scheme? What's the plan for tracking these vaccines and for deciding about the needs for boosters? If the phase 3 trials are only a couple of months long, what does the development plan look like?
So the phase 1-2 studies have interim analyses at the point of peak immunity, which is typically 2 to 4 weeks after vaccination. And those make the decisions to move forward, because you don't want to wait a year before moving forward with something. Similarly, the phase 3 studies will have early looks at the data. I believe that they're powered at 150 events, but there'll be early looks at about 50 and 100 events. So there could be efficacy signals very quickly, and they're designed that way if they are highly successful vaccines. But all of these studies will follow participants out. So it might not be in the news as much, but the phase 1-2 studies actually are full 1 to 2 year studies. And so there will be 1 and 2 year durability data from our vaccine and I think all of the other vaccines. Sure. It's just not the data that's used for decision making up front. But I do think the question of durability is a key question, because if all we can do is protect people for 4 weeks, then these vaccines will fail to achieve their primary objective of global pandemic control. There's no question that durability might be an Achilles heel of some or many of these vaccines, because we simply don't have any data on durability for any of these vaccines yet. And that's a major unanswered question. What we do know is that for the Ad26 Zika vaccine, and that manuscript is under review now, in humans, we showed that with an Ad26 Zika vaccine, that a single shot of that vaccine raised moderate levels of neutralizing antibody titers in all recipients, and those neutralizing antibody titers persisted with no evidence of decline for a full year, which is the length of follow-up of the study. So we don't know, of course, whether that will translate into a COVID-19 vaccine, but at least for Zika, we got neutralizing titers in 100% of recipients with no decline whatsoever for a full year.
It's really exciting data. Okay, we've received a couple of questions about the RBD versus spike protein. So is there any advantage of using RBD versus spike protein as a vaccine target in regard to the potency of the neutralizing antibodies induced?
Well, I think it's primarily a question for the protein-based vaccines. And I think the protein vaccine developers are actively pursuing both RBD fragments as well as full spike. RBD might be easier to produce, it might produce more easily, it might produce at higher levels, but we do know that the majority of neutralizing antibody responses, but not all, are directed against RBD. So there are neutralizing antibody responses against other regions as well. And when we look at antibody titers by ELISA, we routinely see higher antibody titers against the full spike than against RBD. In our own DNA and mouse Ad26 studies, we didn't put it in the monkey Ad26 study, we also made RBD vectors. In the context of our vector platform, then we get lower neutralizing titers with the RBD fragment of the full spike. And whether that's a vector-specific issue of biochemical production, whether that's translatable to a protein-based approach, we don't know. So we're not saying that our results in any way have any implications for the use of purified RBD as a protein-based vaccine, for instance. But at least in the context of these DNA vaccines and vectors, then we think that there is an advantage of using the full spike over RBD.
Yeah, that's helpful. Thanks. We received a couple of questions about the Ad26 for Ebola and just the context of the COVID development for vaccines. So if, so the Ad26 Ebola vaccine that's approved by the EMA as a prime boost vaccine together with an MVA vector vaccine, is this a similar concept required for the COVID vaccine in the Ad26 model? Is that needed as a prime boost combination?
Oh. No. So it'll be a standalone vaccine, similar to, say, the Zika vaccine. So there won't be any boosts. There won't be any heterologous boosts. So the COVID vaccine will be a single shot vaccine, essentially a once and done Ad26, or two homologous shots of an Ad26 of the same Ad26 vector. So those are the two different vaccines that we're looking at clinically. We're not looking at any MVA boosting.
And is there any effect if someone's received the Ad26 vaccine for Ebola? Will that affect their response to the Ad26 COVID vaccine?
That's actually a very good question. So if a COVID vaccine is truly used widespread globally, then it is likely that other vaccines will have to be remade on a different serotype.
Okay, we received a few questions about your study and the study design. Did any of your partially protected and infective sham control animals show pathological sequelae? Did you do necropsies to look?
Yeah. So we did not see any evidence of enhancement of disease. The studies, I should mention, the studies, I didn't include that in my data talk, but there is a number of supplemental figures in the paper addressing that specifically. Although these studies were not specifically designed to show safety, then we did note that in the breakthrough animals, there was never any evidence of enhanced or worsened disease, either clinical, virologic, or pathologic. What we saw essentially was just an abrogation of the protection. We never saw a worsening compared with the sham controls. We also saw that the vaccine induced a pure Th1 and no Th2 responses, and we actually did an analysis of BAL cells, both post-vaccine and post-challenge, and we demonstrated both in cellular composition as well as in pathology sections that there was no evidence of eosinophilic infiltrates. And so I think those are the three main questions people ask.
Yeah, we're getting asked that a lot as well. So someone's commented, Gilead also recently published a paper looking at the correlates of immunity of mild patients and deceased how does your AD26, how does the candidate, the SPP, that top candidate, compare to optimal natural correlates of immunity of convalescent patients? Have other vaccine candidates published similar studies on the correlates of immunity beyond the neutralizing antibody titers?
I'm not aware of any of the other. So now there's, I think, 6. We actually, my student and my postdoc wrote a review, and we decided that there were 6 published monkey challenge studies for COVID-19 vaccines so far, and there's going to be many more in the next few weeks, and maybe more as of yesterday, because I didn't look at the literature yet. It literally changes every day.
It changes every day, sometimes hourly.
But there have been, I would say, at least 6 monkey challenge studies looking at candidate vaccines. And I don't believe that any of the other ones looked beyond binding and neutralizing antibodies into other functions.
Have you tried to clone and sequence any of the virus after challenge to check if there's any mutation?
You know, we've thought about that, but we haven't done that. So we have specimens, and when we have BAL samples, we could do a whole lot of sequencing. We haven't done that. So we haven't looked at the breakthrough viruses.
So adenovirus in humans appears to generate less potent neutralizing responses than subunit vaccines sometimes. Do you agree? Do you think adenos have advantages regarding durability? Do you think there are advantages in the upper respiratory tract infection?
I think it remains to be seen. I think there's a lot of data that remains. So there's no doubt that in terms of straight neutralizing titers, then 2 shots of a RNA or a protein vaccine give higher neutralizing titers than 1 shot of an adeno vaccine. We actually haven't tried the 2nd shot, but we do think it'll probably increase titers by about 10-fold or so. And then in terms of the questions of durability and questions, we do think that a T-cell response is likely necessary for durability, as well as for some mop-up therapy, maybe in the upper airways, if secretion of antibodies doesn't get into nasal secretions all that well. But how that will play out, I think those data have not yet been developed.
A follow-up question to that, you can comment on the transmembrane domain facility, if that facilitates antibody responses. Would access to FEC in lymph nodes be limited?
That's an interesting question. We actually initially hypothesized that a soluble fragment might be more immunogenic, and it turned out not to be the case. So we don't really know the mechanism as to why the transmembrane version of the protein did better than a stabilized soluble ectodomain. So we could speculate, but we don't have data on that.
Okay. Those last few questions actually came from Michael Farzan, who is our next speaker in 2 weeks' time. So it'll be interesting to see. I know his lab is also recently published and put out some interesting data. So it'll be interesting to see his take as well. We are at time. Do you have time for a few more questions, or do you have to rush off? I know your time is valuable and limited these days.
I do have an 11 o'clock call I'm supposed to be on. But
okay, so then we're...
I can take one more question, how about?
Okay, sure. Let's see. Okay, so we received a few questions about the intranasal administration before looking at mucosal immunity. Have you added mucosal immunity to these studies, or is that something?
Yeah. So in our paper, we did show low levels of antibody titers, IgG and IgA in BAL fluid. We actually haven't looked at nasal swabs, just for the reason that we only get 2 nasal swabs, and we use at least 1 for virus, and I've been hesitant to use the last one to look at nasal antibodies. But at some point, we need to do that, because that's an important question.
Yeah, that makes sense. Okay, well, thank you so much, Dr. Baruch, for your time. I know you're doing a lot of these talks and busy in the labs. We really appreciate your time. Thank you so much.
Well, thank you all. I appreciate everyone's input and questions, and everyone be healthy.
Likewise, thank you. And thank you, everybody, for participating and sending in your comments and questions. I invite you to join us 2 weeks from today on the next Global COVID Lab meeting, which is scheduled on August 27th at 10 a.m. Eastern Daylight Time. Our next speaker will be Dr. Michael Farzan from the Scripps Research Institute, who will talk about ACE2, the SARS-CoV-2 S protein, and better RBD-based vaccines. If you're interested in more research on COVID-19, please sign up for the HVP COVID report. In this newsletter, we present highlights and insights from experts all around the globe and highlight the latest scientific articles and data. And lastly, please feel free to visit our website and follow us on LinkedIn. We're going to upload a recording of this webinar there for your review. So with that, I'll say stay safe, wear a mask, and we hope you'll join us again on the next Global COVID Lab meeting. Thanks, everybody.